From the smallest bacterium to the largest whale, life is a symphony of molecular interactions. Molecular biology seeks to understand this symphony by studying the molecules that make up living organisms and how they interact. Just as an orchestra relies on the precise interplay of instruments, life relies on the precise interplay of molecules. These molecules, working together, determine the characteristics of all living things. Understanding molecular biology is crucial to understanding life itself. It allows us to explore the fundamental processes that occur within us and all around us. From the replication of our DNA to the digestion of our food, molecular interactions are at the heart of it all. By unraveling the mysteries of these interactions, we gain insights into the complexity and beauty of the natural world. Imagine the human body as a vast, intricate city. Within this city, molecules are constantly moving, interacting, and changing. They are the workers, the builders, the communicators, and the energy providers. They carry out the essential tasks that keep the city, and therefore the body, functioning. Molecular biology gives us the tools to study these processes in detail. By understanding how molecules interact, we can develop new drugs to fight diseases, engineer crops to be more resistant to pests, and even design new life forms. The possibilities are endless. To understand the symphony of life, we must first understand the instruments atoms and molecules. Atoms are the smallest unit of an element that retains the chemical properties of that element. Think of them as the individual notes in the symphony. Molecules, on the other hand, are formed when two or more atoms bond together. They are the chords and melodies. Atoms are made up of even smaller particles called protons, neutrons, and electrons. Protons have a positive charge, electrons have a negative charge, and neutrons have no charge. The number of protons in an atom determines what element it is. For example, all carbon atoms have 6 protons. Atoms bond together to form molecules by sharing electrons. This sharing of electrons is called a chemical bond. There are different types of chemical bonds, but the most common are covalent bonds and ionic bonds. Covalent bonds occur when atoms share electrons equally, while ionic bonds occur when one atom donates an electron to another atom. These bonds are not static, they are dynamic and constantly breaking and forming. This dynamic interplay of bonds is what allows molecules to interact with each other and carry out the complex processes of life. Think of it like a dance floor. Atoms are the individual dancers, and the bonds are the connections they make as they move and interact. Just as the dancers can change partners and move in different formations, atoms can form different bonds and create a wide variety of molecules. Within our cells exists a universe of biomolecules, each with its own unique structure and function. These molecules are the workhorses of life, carrying out the essential tasks that keep us alive. There are 4 main classes of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, like sugars and starches, are our primary source of energy. They are like the fuel that keeps our cellular machinery running. Lipids, including fats, oils, and waxes, store energy and form the structural components of our cells. Think of them as the building blocks and energy reserves of our cellular city. Proteins are the most diverse class of biomolecules. They are involved in almost every aspect of life, from catalyzing chemical reactions to transporting molecules across cell membranes. They are the workers, the builders, and the communicators of our cellular city. Nucleic acids, such as DNA and RNA, store and transmit genetic information. They are the blueprints that contain the instructions for building and maintaining our cellular city. Each class of biomolecule has a unique structure that dictates its function. For example, proteins are made up of long chains of amino acids that fold into complex 3-dimensional shapes. This unique shape allows proteins to interact with other molecules in a very specific way, like a key fitting into a lock. The diversity of biomolecules and their interactions is staggering. It's like a vast, intricate clockwork mechanism, with each molecule playing a specific role in keeping the entire system running smoothly. Understanding the structure and function of these biomolecules is essential to understanding how life works at the molecular level. Molecules are in constant motion, but what keeps them from flying apart? The answer lies in stabilizing interactions, such as hydrogen bonds and van der Waals forces. These weak interactions are essential for maintaining the structure and function of biomolecules. Hydrogen bonds are like weak magnets attracting slightly positive hydrogen atoms to slightly negative atoms like oxygen or nitrogen. These bonds are crucial for holding together the two strands of DNA, giving it its double helix shape. They also play a vital role in protein folding, determining the protein's specific three-dimensional structure. Van der Waals forces are even weaker than hydrogen bonds, arising from temporary fluctuations in electron distribution around molecules. Though individually weak, these forces become significant when summed over large molecular surfaces, like the interaction between a gecko's foot and a wall. These stabilizing interactions act like a delicate dance, holding molecules together just tightly enough to maintain their shape and function, yet allowing for flexibility and movement. This balance is crucial for life because it allows biomolecules to interact with each other in a dynamic and controlled way. Think of it like a group of dancers holding hands. The grip isn't permanent, but it's enough to keep them connected as they move together. Similarly, stabilizing interactions hold biomolecules together in a dynamic dance that allows for the complex processes of life to occur.